That's my girl, Nana. Nana's out there saying, round 'em up. Okay, well, I'll start with introductions in the meantime. If we can change the slide to PowerPoint, please. Thank you. So only 2 talks left. You guys are such troopers, sticking around. 2 talks and then roundtable, which is nice and casual, and it'll be a nice time to hear questions in a very candid manner. But this next talk, I just want to introduce, you've already introduced to Dr. Ed Weeber, and this is Dr. Jodi Cook. Jodi Cook is a PhD. She's the head of gene therapy strategy for PTC Therapeutics. You have known Jodi now for probably 4.5 years. And Jodi has been an immense contributor to this community, particularly emotionally, for at least me and Paula. So we love Jodi to pieces, and we are so excited for the new endeavor that Agilis has now been acquired by PTC Therapeutics, and they just seem like a wonderful company, and we're very happy to welcome PTC, who's here for the first time, and we're excited to hear from Ed and Jodi about the status of their gene therapy program for Angelman syndrome. No, it's a PowerPoint.
Bring it to full screen.
Yeah, I'll do full screen. Okay, that should work. Good.
Great, thank you, Allison. And I know you hate to steal everybody's thunder, but if you look at my notes, you kind of hit the first 3 points on it. But I'll repeat it anyways. Thank you all for sticking around late in the evening. My name's Jodi Cook, and like Allison said, I've been in this community now for a little over 4 years, and I'm privileged to speak to you, and I really enjoy meeting and talking to you at these events and throughout the year. I am currently the head of gene therapy strategy at PTC Therapeutics. Previously, I was at Agilis, and this last summer, Agilis was acquired by PTC. Agilis has programs in various, or had programs in several different therapeutic areas of gene therapy. PTC is a global rare disease company. The company only focuses on rare disease. The company has 2 marketed products in Duchenne muscular dystrophy, and we have a worldwide footprint. We have 500 employees. So when PTC approached Agilis, management met the management at PTC, understood who they are, understood what their values are, and we championed this acquisition through our board of directors. So we believe that now as PTC, we're even better equipped to bring these therapies to children. In the gene therapy space, our lead indication is an AADC deficiency. And I think 2 years ago, I showed you all videos of what's possible in gene therapy. Now that therapy is being prepared to be filed for an approval with the US FDA next year. So we're very excited about that. And now as part of PTC, we're going to aggressively move our Angelman program forward. So I have brought other colleagues with me to this meeting. Thank you. So I know everybody worries about it, and that's what happened, and we all do. Somebody gets acquired and then things stop and go away. I promise you that is not the case here. If anything, they said, the deal is closing, don't stop, we're going faster. And now we have 500 colleagues to make that happen. So it's no longer just a handful of us doing everything. We have a large number of people involved. And to that, I have my colleague, Mary Frances Harmon, who's here with us today and is staying through Saturday. So please introduce yourself to her. She's head of our patient advocacy and corporate communications. And Dr. Joe McIntosh was through here today and yesterday as well as part of the ABOM. So things are moving forward, and I'm excited to say that we're continuing our great collaboration with the University of South Florida and Dr. Ed Weeber. We are actively involved in the ABOM, and we have been since the very beginning. And I also want to say, as Megan Cross had mentioned earlier, the patient registry is absolutely critical, not just for PTC, but for all of the companies and all of the academics and all of the registries, or in all of the trials that are upcoming. So not just for trials, but for future improvements on therapies once we get the first ones approved. So please register for that. It'll help the community, it'll help all of us. So I'll stop now and turn over to talk about our great science that's going on with Dr. Weeber. Thank you.
It's great to see everybody's still here, and there's a lot of interest still out there in the with all of you, and I appreciate you staying. This is going to be quick, but I want to just kind of give you an update, and we've talked about gene therapy before, and Jim Wilson gave a really fantastic talk about gene therapy, and we're basically doing the same thing. We're looking to replace the UBE3A gene using an AAV. This was a proof of concept a long time ago. I think we published this paper in 2011. We were the first ones to kind of show that AAV could be used. We picked up one of those seeds that Jim Wilson left, and it was kind of a brand new technology. I don't know where Jen Daly is. Is she still out there? Oh, there she is, way over there. So Jen Daly was a graduate student in my laboratory. She was looking for something to do her dissertation on, and we came up with this idea. It was a brand new technology. We thought we were coming right off of the CAMK2 paper where we reversed all the neurological defects in the mouse model. This was a collaborative work with Ipa Elgersma, and we decided then that, and we realized then that this was not developmental. That everything was still there, that the brain, at least in the animal model, all the connectivity was there, it was formed right. So what would happen if we replaced the protein? And back then we were very hippocampal focused. And so what we did was we developed the AAV, and we did a direct injection into the hippocampus. And so you can, let's see if I can pull this up. So Allison kind of talked about this, brown is good. So this is a cross-section of the hippocampus, and we have an antibody that recognizes the UBE3A protein. And you can see here is a wild-type animal, a non-genetically altered hippocampus of an animal. Here's an AS, and here's an AS that we've given the AAV. So all the neurons now are rich with the UBE3A protein that was delivered there by the AAV. Okay, and we saw a great recovery. We saw recovery in the learning and memory defect, and we also saw recovery, at least also almost a full recovery of the synaptic plasticity defect, which I talked about earlier. So last year I gave this slide and talked about how difficult it was, and looking at all the different combinations. Now when we talk about AAV, there's a lot of different serotypes of AAV. There's a lot of different types of AAV out there. There's 3 different isoforms of UBE3A. We talked about different routes of administration, and Dr. Wilson talked about that. Do you do intrathecal? Do you do cisterna magna? So we're thinking about all that. There's promoters. So you just don't put the gene in there. You have to put a little piece of DNA in front of it to turn that gene on. And there's a lot of different promoters out there as well. So when we started calculating all these different combinations up, you have 1,800 different possible combinations of this. And so that was overwhelming. And so we really had to kind of call it down and say, okay, this is what we're going to look at. We had a kind of ace in the hole. So Kevin Nash over there is at the University of South Florida. Yeah. So Kevin is an expert in AAV and being able to clone these genes, get them packaged, put them in the AAV, and then give it to us so that we could do our cool work. Now, just a really quick story about Kevin the first time that I met him. This is really difficult stuff. And so, when I wanted him to help me, somebody said, look, it takes a lot of time to do this. There's a lot of people that are trying to use this technology. He was doing all this work in Alzheimer's disease, and so I went to talk to him. I figured he was probably doing maybe 2 of these a year, and I wanted to know when I could get put into that slot so that he could help me. And so he pulls down this book, and he starts flipping through all of the AAVs that he had produced that year, and there was 20 of them. And I was like, oh, wow, that is really incredible. And I said, you did all this in 1 year? And he was like, oh no. And he pulled down another book. And he was like, that book was filled up. And he said, I did all these too. He's a machine. And it really, really helped us with all those combinations. So where are we now? So now we've really culled everything down. We're looking at just a couple isotypes. We already know the isoform that we're using. We're looking at a couple of the promoters, and we're really kind of concentrated, at least now, on one route of administration. So that 1,800 possible combinations is now down to about 4 possible combinations, and we're focused on that right now. So one of the questions that I always get is, the gene is huge. It's this big gene that's in the on the chromosome 15. How do you get it to fit into an AAV? And so you guys are probably getting pretty good now at understanding the biology of this, what the gene looks like, what the genome looks like, and this transcription and translation. So I have a little bit of a graphic here to help especially the new parents that are new to the science in here. So you got to think about the whole genome as a big recipe book, right? And so in all of those letters is really the recipe for specific proteins. So you can look at this long string of letters here, and in there you can see a cup of flour, 2 cups of sugar. I know you're thinking I like my muffins really sweet, but this is really the recipe for that single muffin, right? Okay, it's the same thing with your genome. You have DNA that's intermixed into DNA that doesn't really make any sense, but with transcription, it all comes together to form the recipe for the protein, in this case UBE3A. So what we've been able to do is figure out that mRNA and just use the DNA that encodes that gene. And interestingly, and I think really importantly, it just is small enough to fit into an AAV. If it was a little bit bigger, I know, if it was a little bit bigger, it wouldn't fit. And let me tell you, the first couple times that we did this, we couldn't quite get it to fit in there, and Kevin was the one that figured out how to actually get it in the AAV so that we can inject it into the mouse. Now, I know that Dr. Wilson talked about this earlier. Just to re-emphasize this isn't a virus that we're using. Okay, it's an adeno-associated virus. Viruses are really good at seeking out a specific cell, going in and turning that cell into a virus-making machine. I mean, this is what causes an infection. So when we're delivering this gene, we're not causing an infection. I've actually seen PhDs come up and use the word infection. I don't want you to think that this is an infection. But the adeno-associated virus is it's very, very good at recognizing specific cell types. Okay, so the gene's got the recipe. Okay, the adeno-associated virus is just the envelope with the right address, neurons in the brain, neurons in the CNS, where we want to deliver the recipe to make our protein. And so Dr. Wilson showed this. We've done this in the lab a couple times. We actually use this as a control for looking at distribution. But this is a hippocampal section here. And these are all the neurons that are glowing with green fluorescent protein that we packaged into that AAV, right? So we can kind of look at this distribution, but we've gone beyond that. Now we're starting to look at distribution. How do we get it everywhere in the brain? And this kind of goes to that 90-10 that Dr. Wilson was talking about, right? So how do we get it everywhere? And we think that just focusing on the hippocampus, which was great for a proof of concept, and we were showing that it had amazing effects in the mouse, but when we're starting to think about clinical trial, 2 things changed, and I think that Jodi and Mark and those associated with PTC now kind of keyed us in on this. Number 1, you should start using the human gene when you're doing these studies, which we didn't really think about. And number 2, we really need to think about distribution. How can we get it everywhere in the brain? And so this is a big question. Do we target a specific area, or do we try to get it as much as we can, more than that 10%? And so we started looking at lateral ventricle injection, hitting the ventricles, which are the spaces in the brain that cerebral spinal fluid kind of flows through. So if you hit that one place, then the virus can kind of go everywhere, get it in as many neurons as possible, and hopefully you'll see that greater effect. And if you look down here, you can see this is prefrontal cortex, striatum. This is the outer cortex, hippocampus, cerebellum. This is our GFP injected, so you shouldn't see a whole lot of UBE3A expression because we're not putting in the gene there. Here is our UBE3A, human UBE3A ICV injection in an AS mouse, and you can kind of look at the difference between a wild type. So you can see all of this brown that's being produced in the hippocampus and in the cortex and different areas of the brain. That's all UBE3A that's being produced in those neurons. So it really tells us that if we do ICV injection, we can get it everywhere, and that's what we're focusing on. So what are the outcomes? And we've seen a lot of these different outcomes. One of the things that we see is a really nice change in learning and memory, okay? And so there's a whole bunch of data here. I don't want you to concentrate on this. I got a little film here. So some of you haven't seen water maze before, but I'm going to show it to you. This is a mouse in a water maze. This mouse has been put in for the first time. You can see he's completely clueless. You can see the platform right here. Okay, here's a wild type mouse, looks the exact same way. They're just kind of swimming around aimlessly. Sometimes they'll find the platform, sometimes we have to train them for the platform. But we do this 4 times a day, day after day after day. So here's the AS mouse on day 5. Now there's cues around that pool, and this mouse will look around, kind of orient himself, all right, and then he'll swim to the platform. And here's a wild type mouse on day 5 as well, and you can see he can do this actually a little bit better. Okay, so now what we do is we test the mouse. So that's the training part, that's the learning part. So now we're going to test how well it remembers where that platform was. And the way that we do this is we remove the platform. Okay, so the platform isn't there. So now we're going to see how much time that animal spends where it learned where the platform was by using those visual cues. All right, oops. Let me go back. Okay, so that's our wild type, day 5. Here's the probe trial. Here's the memory trial that we're going to do. And this is the AS mouse. So again, we've taken the platform out. You won't see it under the water anymore. Okay, so he should spend a lot of time over here. And you'll see he's forgotten where the platform was. So after a little bit of time, he doesn't remember where the platform is. He's just randomly searching around. And now we'll show you what a wild type mouse after day 5 on a probe test looks like right now. And you'll see the difference here. Okay, so he's orienting himself and then he goes, oh, where's the platform? It should be right here. And so it starts crossing back and forth over where the platform was. He can't get on the platform, but we can see how much time he spends in that little area. And this is where these heat maps come in. Okay, so we do all this trial on a number of different mice, and then we see how much time they spend in the trained quadrant. Okay, so here's what a wild type looks like. So here's what the platform was. Here's what our GFP injected AS mouse looks like. So it's all over the place. It doesn't spend as much time here. And here's our AAV injected. So it remembers. It not only was trained and could learn, but it also remembered where the platform was. So this is a really nice outcome measure. We can see that with this injection and this distribution, we get really good recovery, learning, and memory. And this is just one of the learning and memory tests that we've done. I talked about this earlier, and we've gone over this a couple times with synaptic plasticity and how important it is, this cellular correlate to learning and memory. It's in the hippocampus. We take the hippocampus out, and then we can stimulate and record across the synaptic connections. And we see that in our AAV-injected mice, we've recovered completely the LTP defect that we saw in the AS mouse. And the AS mouse is in green down here. Our wild type is up here. So we see more plasticity, more communication across those synaptic connections in our AS mice that we've injected with the AAV, come right up to wild type levels. Now the interesting thing, it's a little subtle, but the interesting thing about this is we actually see better recovery of LTP when we did the ICV injection than when we did the direct hippocampal injection. So what does that mean? Well, what we think it means is that when you get better distribution of the AAV and better distribution of the protein, you have globally the brain is just working better and it makes the hippocampus work better. All right, so this is really nice, nice data that we're getting for both the behavioral part of the thing and the physiologic part of the thing. So we have now started to transition from the mouse to the rat, and you've seen a lot of that data today. Things like route of administration, distribution, biomarkers, correlate expression, rescue. I mean, this is all really nicely done in the rat, and the rat is a great model for this. So I'm going to change the people that weren't on ABOM yesterday. Biomarkers are a huge thing. Okay, how do you determine gene replacement efficacy at a genetic or molecular level? Well, in the animal, it's easy. We inject with the AAV, we wait 5 weeks when the protein starts working and we do the behavior. We just take the brain out and look at it. Okay, yeah, that's pretty cool. But we're talking about a clinical trial now. All right, so this idea of biomarkers, how do you tell it's actually working? That 90-10 that Dr. Wilson talked about earlier, I mean, how do you determine that? Okay, so how would you do a non-invasive protein detection? We can't do it in blood. And how would you quantify global gene replacement expression? So how do you determine how much expression is happening in the brain by a biomarker that you can look at later? How do you know it's actually working? So this is something that the FDA is really concerned about, right? So if you're injecting an AAV into the brain of a child with Angelman syndrome, how do you know it's actually making the protein in those neurons? So this idea of a biomarker is really important, and it's something that we've focused on. So I've got a couple different things that are going on here. So I know that you guys have seen these blots before, and I'm not exactly sure if everybody knows what those blots are. It's a Western blot analysis, and we can do polyacrylamide gel electrophoresis, where we put in proteins here, we run them out, and the gel is actually kind of a matrix. So it separates out the proteins by size. So the little proteins go through quick, the big proteins kind of hang out, and then we use an antibody to probe that protein blot. All right, and if we use an antibody for UBE3A, we should see one little band there. So when you see these pictures of these different bands, that's what you're looking at. Okay, you're looking at UBE3A protein that's been run through a gel, and there's 1,000 different proteins there, but the antibody just seeks out that one protein. This is what a Western analysis looks like. And in the hippocampus, cortex, and cerebellum of our rat, we see that there's a reduction. We can look at knockouts here. Cerebral spinal fluid, when we looked at this, AS rats were reduced. Okay, we were lucky enough to be able to get a CSF sample from a patient. This was an 11-year-old female AS patient with a molecular diagnosis for AS. And when we look at this, so this is our recombinant, this is our wild type, this should be neurotypical, not wild type, in our AS CSF here, and you can see that it's significantly reduced. So we have a potential biomarker, but it still begs the question of is this neuronal derived? Is the lack of the UBE3A in the CSF due to the lack of the protein in the neuron? So we can do this in a couple of different ways, and I think Dr. Baudet talked about topotecan, which can turn on the paternal allele. So this is one of the things that we did. We did an intrathecal injection of topotecan, turning on that paternal allele using this method. And when we do this, sure enough, what we can see is an increase in UBE3A protein in the CSF. This is cool, all right? Turn on the paternal gene, you can see the increase in the biomarker in the CSF. You don't have to take the brain out to look at it anymore, okay? All right, so what about gene replacement therapy? Now here, we're turning on the paternal allele. Will this work for what we're trying to do? A gene replacement therapy using an AAV. All right, so this is recombinant, and yes, you can buy recombinant UBE3A. It's really expensive if you want to buy some of it, but you can order it. It's just pure UBE3A, so we can run that on our gel. This is kind of our standard. Okay, so here's a knockout. You shouldn't see any UBE3A at all. We don't. Here's our AS. You see a kind of little tiny bit. It's coming from the paternal allele. Here's our wild type, so nice protein band here. Here's an AS mouse that we've treated with our AAV, and now it's there in the CSF. All right, so it doesn't matter whether you're trying to turn on the paternal allele or whether you're using a gene replacement therapy, we have our biomarker. I can't tell you how huge this is. This is a huge check that we put on the side of this saying we have a biomarker now that we can use. We don't have to do anything invasive to see whether our therapy is working, regardless of what that therapy is. So we also are looking ahead and addressing things like overexpression, which is a concern. So if you're using an AAV, you're not just turning on the paternal allele. You're using an AAV and you're increasing with an exogenous gene. So is there a worry about overexpression? Should we be worried about this? Is there consequences, and how can we test this? Well, as a scientist, this is really easy. Okay, we're getting our AAV, we're putting it in our AS mouse, and we're seeing increase in protein. Let's just do this in a wild-type mouse. That wild-type mouse is already at 100%. All right? So this is exactly what we did. We put our AAV in, human AAV. We injected a wild-type mouse, and we saw that we could increase levels to about 130%. That's interesting in itself because, Megan just showed that in the non-human primate, they could get it up to like 200%. Even with the AAV, we're not getting it up that far. This is a fairly low end, so we're still doing this work. But we are above 100% in those neurons. Okay, so what happens to the changes in behavior? What are those outcomes? This is our spatial learning and memory. So this is our, the water maze. We don't see any changes there. Dr. Alberini talked about contextual fear conditioning. We don't see any change in associative learning and memory. Novel object recognition, which is another learning and memory test. We don't see anything there. We don't see any change in anxiety, slight change in social interaction. They actually become more engaged with a mouse. I know some of you are saying, oh my gosh, my Angelman child is going to be more engaged. This is a mouse model, but we were looking to see if there were any changes that were detrimental to the behavior of the mouse, and we don't see anything at all. Okay, so we're already thinking ahead on possible things that the FDA would say, and that's one of them. What about overexpression? And I know we're trying to get to that, above that 90-10, but if we get to 110, should we be worried about that? And from these animal studies, I would say no. So in conclusion, I think it's clear. All I can think of is, my kids running around the pool. I tell them, don't run around the pool or you can't swim. And so they walk as fast as they can. It seems like we're all walking as fast as we can. We're trying to be as safe as possible, but this you can really see the rush now, and things are moving very, very quickly. But I think from multiple different laboratories, multiple different tests, multiple different investigators, this is a treatable disorder. And I think we need to be cautious, but hugely optimistic, and I think Dr. Baudet is absolutely correct on that. AAV technology coupled with new animal models, like the new rat animal model, we think will provide a really kind of safe and well-studied approach when we start thinking about human treatment. And we've already thought about these things like biomarkers and overexpression. Identifying future questions and preparing for Angelman syndrome patient enrollment will certainly facilitate and expedite clinical trial when we get there, and we will eventually get there. I think that graphic that Allison showed at the very beginning, it's marching forward. You can't stop it. Nobody can stop it now, okay? And it's multiple companies, multiple people, multiple investigators. We're just one of those, but it clearly is moving forward, and I'm excited every day about how quickly we're moving forward as well. There's some people I need to thank. This is Hayden Green. She was the one that I gave the CSF sample to from the AS patient. I gave her that vial, and I was like, this is the only one in existence in the world. If you screw it up, I'm going to kill you. And she came out with some really good Western analysis. And I showed at the ABOM meeting, we actually did an activity assay on that CSF as well, so that we could look at this in a couple different ways. Mindy Peters is she's been with me for a number of years. She's done all the behavior in the mouse model. Jen Daly, I've got to thank, even though she's not in this picture and she's been gone out of my lab for a number of years. We got all this going, and that was part of her dissertation that she got her PhD. And with that, I will take any questions. Thank you.
Thanks very much. I was heartened to see those slides about overexpression not showing any deleterious effects. I am aware, though, of a 2011 study by Stephen Smith that showed overexpression of the UB3A gene caused some autism-like traits, and I was just curious on your observation of that study and your previous comments before.
Yeah, so that is in the interstitial duplication. All right, so this is a duplication of that 15q11 through 13 region. And this has actually gained a lot of attention. There's an animal model now. It really shows a lot of interesting alterations. And I don't know if you saw that sociability was one of those things that we looked at. So this is a really strong genetic correlate to severe autism. But really the question is, why? Is it one of those other genes that Dr. Baudet talked about? It could be overexpression during development. So we're not trying to do that. We're not trying to treat, in utero. We're treating after birth. So there's a lot of kind of questions about that. That was a concern, and that's one of the reasons why we did that work. But it's still unclear why that interstitial duplication causes the phenotype that we see in the animal model or in the human condition. But I think with these studies, I was asking Megan about the non-human primate too. I think as we're moving forward and closer to clinical trial, this will be something that we're thinking about. We're hoping, and you saw the talk earlier, we're hoping to get close to that 100%, but we always have to be concerned, or at least addressing the possibility of getting over that, especially in some neurons. But if we can get close to 100%, that would be great.
It just leads to the broader question of parents with children with UPD and ICD about how worried we should be potentially from any potential exclusion from trials where you have that gene expression, be it turning on the parental allele or from reintroducing that gene?
Oh, yeah, that's a tough question. I mean, I think once we get closer to clinical trial and those inclusion-exclusion criteria, and we have more scientific data to make that determination, it's probably a more answerable question.
Great. Why don't we save the rest of the questions for the roundtable, because Ed and Jodi will both be at the roundtable.
Thank you very much.
Thank you.